Welcome. In this video, we will explore the classification of gram-negative bacteria based on their shapes and staining characteristics from cocci and rods to helical and filamentous forms. Gram-negative bacteria based on the morphology is classified into cocci and rods. First, gram-negative cocci, which include Neisseria, which appears as a paired cocci as shown in this picture. Next is Coccobacilli. It can appear in pairs or in clumps. Some Coccobacilli like Moraxella bovis appear fat and in pairs, while Acinetobacter shows rods that often resemble Coccobacilli. Gram-negative rods. It is further classified based on the Ziehl-Neelsen staining into the Ziehl-Neelsen positive and Ziehl-Neelsen negative. First, we look into the Ziehl-Neelsen positive Coccobacilli. First is Brucella. It forms small clumps and grow on agar media, while Chlamydophila psittaci also form clumps but cannot grow on standard laboratory media. So if you are preparing a smear from the suspected organ sample and if you are getting a Coccobacilli that is gram negative and also Ziehl-Neelsen positive, just grow them in an agar. If it is growing means it is Brucella. If it is not growing means it can be Chlamydophila psittaci, but we have to do further testing. Next is gram-negative rod, that is Ziehl-Neelsen negative. Under that, so many groups are there. First one is medium-sized rod. Bacteria like Enterobacteriaceae, Pseudomonas, Aeromonas, Bordetella, Lactobacillus, Haemophilus, Pasteurella, Alkaligenus, and anaerobic bacteroides all are gram-negative, Ziehl-Neelsen negative rods. These bacteria are important pathogen in both humans and animals. Next, under gram-negative rod, that is Ziehl-Neelsen negative, is curved rod. So it will look like curve shape. The bacteria that is coming under this is Campylobacter and Vibrio species, which are easily distinguished by the unique curved morphology. Next, under gram-negative rod and Ziehl-Neelsen negative bacteria is the helical or the spirochetes. The helical or spirochete bacteria include Leptospira, Treponema, Borrelia, and Serpulina. These are slender spiral-shaped organisms often requiring special stains or dark field microscopy for its visualization. The Serpulina and Treponema, it is visualized by gram staining and DCF stain. Borrelia, you have to go for Giemsa stain. Leptospira, it is very thin, so it won't stain with the gram stain. So we have to go for dark field microscopy. Next, gram-negative rod, that is Ziehl-Neelsen negative, is the Filamentous gram-negative bacteria, which include Fusobacterium species, which are anaerobic and often appear thread-like under the DCF staining. In summary, gram-negative bacteria can be classified by morphology into cocci, Coccobacilli to rods, curved forms, helical spirochetes, and filamentous anaerobes. Understanding their shape and staining characteristic helps in rapid identification in the lab.